Instead of doing experiments on humans, scientists can often answer many of their first research questions using animal models or human cells. Human cells are especially useful for understanding whether the lessons we learn from studying animals are also true for people. They are also very useful for studying human diseases, especially cancer. What is common about all types of cancer is that they result from uncontrolled cell growth and proliferation in the body. Because cancer cells grow too easily, it is often possible to continuously maintain the cells from many human cancers in a culture dish. Normally, the cells are grown at 37 degrees Celsius in a liquid medium that has all the necessary growth factors and nutrients to keep them alive. Cell lines starting from human cancers have been grown in laboratories around the world for many years. Up to now, thousands of cancer cell lines have been made available for research, and each has its own characteristics. When scientists study cancer cells, they usually want to understand why the cells keep growing and how to stop the process. In order to answer those questions, scientists must first understand the molecular details of how cells proliferate. At ICOB, the East Lab studies how molecules interact during cell proliferation and division. The somatic cell cycle contains 4 phases, including an initial growth phase, a DNA synthesis phase, a second growth phase, and mitosis. The progression from one stage to the next is highly controlled. During mitotic cell division, the nuclear material is organized into chromosomes that are segregated into 2 daughter cells, and the success of this process relies on the precisely controlled assembly of the mitotic spindle. This topic is one of the major research areas in Dr. Yi's lab. The mitotic spindle is mainly made up of microtubules and a pair of complex structures called centrosomes. The 2 centrosomes localize to each side of the cell, where they nucleate and anchor the microtubules that attach to chromosome in order to properly segregate them. Numerical or structural defects in centrosomes disrupt their essential functions and usually lead to mitotic spindle abnormalities and cell death when the cell attempts to divide. Disrupting centrosome functions could therefore be a way to stop cancer cells from growing. The East Lab members mainly use microscopy approaches, such as immunofluorescence, confocal imaging, or super-resolution imaging to examine the 3D structure of the mitotic centrosome. Their work has demonstrated that centrosome structure in genetically or pharmacologically challenged cells may be severely disrupted compared to normal cells. This defective centrosome structure results in abnormal mitotic spindle and reduced cell proliferation. Work such as this is essential for understanding how cells divide, and the knowledge gained may lead to new ideas for cancer treatment.